Hi, I'm Jenna Herbst, and I will be talking on ocean depth on carbon flux. One of the species of interest is the bivalve species, which can be known as mollusks or clams, oysters, organisms of that sort, and also gastropods, which can be seen as snails. Some of the main takeaways from this research are that both of the species had a declining carbon flux with increasing depth. This means that the initial hypothesis was supported, and the bivalve species was significant. However, the gastropod species was not significant. There was a greater variation of carbon flux towards the surface for both species, and both of the papers account for this with different factors such as body size, food availability, temperature, and metabolic activity. And another takeaway is that carbon cycling depends on depth, which needs to be researched further in order to understand the effects from climate change. With the future of climate change, this can affect carbon cycling in marine ecosystems, which can greatly impact trophic levels. From figure 1, we can see that log mean flux was plotted on the y-axis and depth was plotted on the x-axis. The bivalve species are the color blue and gastropoda are colored in red. And both species had a declining slope for each of the line, which thus shows again a declining carbon flux with increasing depth. For figure 2, the anti-log was taken in order to better account for this variability within the surface depths. And we can also see that there was still this declining slope, which further supported the hypothesis. In regards to why it matters, trophic interactions may be altered, and this can lead to primary production also being altered, affecting many species. Climate change can also lead to a warming climate that will also affect carbon flux. With carbon cycling, the ocean is very important for absorbing atmospheric carbon, and the marine health of these species is very important to consider in order to see how their marine life will change in the future. From paper 1, the first 50 carbon flux data for each species was taken, so this was a total of 100 data points. And like I had mentioned, that the depth was plotted on the x-axis and log mean flux was plotted on the y-axis. And the anti-log was taken, or just multiplied all the carbon flux data by 10 in order to obtain figure 2. A linear regression test was performed in order to obtain the r-squared value and p-values. In this paper, there was no way to account for any error that could have occurred during the experiment. Paper 2 also had the first 50 carbon flux data, so a total of 100, plotted similar ways like I'd mentioned before. A linear regression test was also done to obtain the same values, and there is no way to account for error in this paper as well. And thank you for listening.